Good morning, good afternoon, and good evening. I will be discussing the metabolism of a very biochemical important molecule, nitric oxide. By the way, I am Dr. Noel Martin Bautista from the Department of Biochemistry, Molecular Biology, and Nutrition of the USC Faculty of Medicine and Surgery. This will be our roadmap for this presentation. First, we will explore the different physical characteristics of nitric oxide. Secondly, we will particularly look into the free radical chemistry of nitric oxide and what makes it a reactive oxygen species. Next, we will briefly enumerate the various and diverse physiological effects of nitric oxide responsible for its biochemical importance and versatility. Following this, we will examine the specific mechanism of action of nitric oxide. We will then proceed to the biosynthesis of nitric oxide, concentrating on the nitric oxide synthase complex and how nitric oxide is synthesized from an amino acid. After this, we will look into the different isoforms of the nitric oxide synthase, leading to its three different classifications, nNOS1, nNOS2, and nNOS3. And lastly, we will study several clinical aspects of nitric oxide immediately following the discussion of each specific isoform of the nitric oxide synthases. Without further ado, let us now look more closely at the structure of nitric oxide. To start, nitric oxide is also chemically known as nitrogen monoxide and abbreviated as NO. At room temperature and atmospheric pressure, the small free radical is a highly reactive colorless gas with low solubility in water. The high diffusion rate in biological systems allows the molecule to reach the targets before degradation. It has been found to occur naturally in various cell types and parts of the body. Cells in the brain, the liver, the pancreas, the gastrointestinal tract, the blood vessels, and the penis can synthesize it. It is a signaling molecule in cellular communication, a neurotransmitter, and a cellular toxin. The molecule is both a reactive oxygen species and a free radical owing to the unpaired electron of nitrogen, which is responsible for its property of paramagnetism. However, as a free radical itself, it is almost unreactive as compared to other oxygen radicals. It decays within seconds after its synthesis if left unbound in solution because it reacts with either molecular oxygen or superoxide, thus its very short half-life. The molecule possesses a small dipole moment because of the similar electronegativity of oxygen and nitrogen, making it essentially hydrophobic. Nitric oxide reacts rapidly with atoms or molecules that are also paramagnetic, predominantly with molecules that have orbitals with unpaired electrons, which are typically either free radicals such as superoxide and hydroxyl ion, or transition metals like heme iron found in hemoglobin, myoglobin, and cytochromes, and metals such as iron, copper, or manganese. As previously mentioned, nitric oxide reacts rapidly with atoms or molecules that are also paramagnetic. One important example is the strong reaction of nitric oxide with superoxide to form peroxynitrite. We will see the significance of this later in the presentation. Here is a brief list of the physiologic effects of nitric oxide. We will be discussing this in greater detail when we go to the different isoforms of nitric oxide synthase. First, nitric oxide is a potent vasodilator which plays a very important role in the regulation of blood pressure. Secondly, it can also act as a neurotransmitter in the brain and peripheral nervous system. Thirdly, nitric oxide is also a signaling molecule involved in cellular communication. Additionally, nitric oxide is a cellular toxin that acts by destroying pathogenic microorganisms, playing a vital role in the inflammatory response. It likewise plays a vital role in the relaxation of skeletal muscle. And lastly, nitric oxide inhibits adhesion, activation, and aggregation of platelets. Let us now discuss the mechanism of action of nitric oxide. To start off, it is worthwhile to remember that the physiologic effects of nitric oxide are mediated through the activation of soluble guanylate cyclase to produce cyclic GMP from guanosine triphosphate or GTP. Guanosine 3', 5' cyclic monophosphate, or otherwise known as cyclic GMP, or simply as cGMP, is a second messenger molecule that modulates various downstream effects, including vasodilation, renal phototransduction, calcium homeostasis, and neurotransmission. cGMP mediates these effects via activation and alteration of specific ion channels, association with cGMP-dependent protein kinases, and interaction with phosphodiesterases, as we will see later on. As mentioned, the physiological functions of nitric oxide are mediated through activation of soluble guanylate cyclase to produce cyclic GMP from guanosine triphosphate. Guanylate cyclase is a heterodimeric, heme-containing protein that converts GTP to cyclic guanosine monophosphate, or cGMP. It is composed of alpha and beta subunits. Cyclic GMP, as we have learned, is a second messenger involved in many signal transduction cascades. As we can see in this illustration, the inactive form of guanylate cyclase has a penta-coordinate heme, bound by histidine on the beta subunit. Nitric oxide now binds the 6th ligand to form a hexa-coordinate heme and causes breakage of the heme-histidine bond. The breakage of this heme-histidine bond now yields the active penta-coordinate nitrosyl complex of soluble guanylate cyclase. Activation of soluble guanylate cyclase causes up to 400-fold increase in the rate of cyclic GMP formation. On the other hand, an enzyme exists for the breakdown of cyclic guanosine monophosphate, which we know is responsible for most of the physiologic effects of nitric oxide. This enzyme is phosphodiesterase, abbreviated as PDE, particularly PDE5, which is specific for cyclic guanosine monophosphate, hydrolyzing it to 5' guanosine monophosphate, or GMP, thus inactivating it and turning off the signal. Moving on, let us now discuss the biosynthesis of nitric oxide. The biosynthesis of nitric oxide is catalyzed by a group of enzymes known as nitric oxide synthase, or simply known as nNOS. Nitric oxide synthase is a group of enzymes responsible for catalyzing the production of nitric oxide from the amino acid arginine. nNOS is a family of heme and flavin-containing enzymes that catalyzes the synthesis of nitric oxide through two serial monooxygenase reactions analogous to cytochrome P450 systems. To remember, monooxygenation is the incorporation of a single atom of molecular oxygen into a substrate and the other oxygen atom being reduced to water. To continue, nitric oxide synthase is a dimeric enzyme that consists of two major domains, namely the oxygenase domain and the reductase domain. Let us now discuss the first domain, the oxygenase domain. This domain contains the following components and their functions: a heme moiety resembling that of cytochrome P450, which acts as the enzyme's catalytic site, a substrate binding site, which is the location for the binding of the amino acid arginine, a tetrahydrobiopterin binding site, which is vital for the process of electron transfer, and lastly, a tetra-coordinated zinc atom for the structural integrity of the oxygenase domain of the enzyme complex. Moving on, another domain is the reductase domain that contains the flavin nucleotides, flavin mononucleotide, or FMN, and flavin adenine dinucleotide, or FAD, which are involved in the transfer of electrons within the domain. It is worthwhile to note that this flavin-containing domain structurally resembles that of cytochrome P450 reductase enzyme complex. Additionally, the nitric oxide synthase contains a calmodulin binding site. This site connects the oxygenase and reductase domains. Functionally, this site serves as a site for calmodulin attachment required for the conformational changes needed for the activation of the whole nitric oxide synthase complex. Let us now look more closely at calmodulin. Calmodulin, an abbreviation for calcium-modulated protein, is a multifunctional intermediate calcium-binding messenger protein expressed in all eukaryotic cells. It interacts with many other proteins in the cell and acts as a regulator or effector molecule in a wide variety of cellular functions. It is an intracellular target of the secondary messenger calcium plus 2 ions, and the binding of such ions is required for the activation of calmodulin. Once bound to calcium plus 2 ions, calmodulin acts as a part of a calcium signal transduction pathway by modifying its interactions with various target proteins such as kinases, phosphatases, or in this case, the nitric oxide synthases. At intracellular basal calcium levels, calmodulin is not bound to the enzyme. It requires an influx of calcium ions to raise the calcium ion concentration, thus increasing the binding of calcium to calmodulin. The calcium-calmodulin complex binds to and activates the nitric oxide synthases. Calmodulin binding causes a conformational change in the enzyme, potentiating electron transfer between the flavins and allowing productive heme reduction. However, in the absence of calmodulin binding, heme reduction occurs only very slowly and does not support nitric oxide formation, and the transfer of electrons between FAD and FMN is also slow. To continue, the general enzymatic reaction of nitric oxide synthase involves the sequential monoxygenation of arginine to nitric oxide and citrulline. This is primarily initiated by the binding of calmodulin to the calmodulin binding site domain of the nitric oxide synthase in order to open the gate between the reductase and oxygenase domains, initiating the biosynthesis of nitric oxide by the nitric oxide synthase complex. With the gate now open between the reductase and oxygenase domains, the electron donor NADPH can now donate 2 electrons to the flavin adenosine dinucleotide or FAD moiety, which in turn reduces flavin mononucleotide or FMN. The FMN reduces the heme ion to its ferrous form, to which oxygen can now bind to initiate the monooxygenation processes. This schematic diagram illustrates the general enzymatic reaction within nitric oxide synthase. Electrons are transferred from the electron donor, NADPH, which in turn reduce FAD and FMN subsequently until reducing the heme ion to its ferrous form, to which oxygen can now bind for the synthesis of nitric oxide from arginine. Let us now look more closely into the enzymatic reaction catalyzed by nitric oxide synthase, which involves the sequential monooxygenation of arginine. In the first step, arginine is oxidized to the soluble intermediate N-hydroxy-L-arginine. One oxygen atom of the molecular oxygen is incorporated to the amino acid, and the other oxygen is reduced to water. In the second step, however, N-hydroxyl-L-arginine is oxidized to nitric oxide and citrulline with the reduction of an atom of oxygen to water. The source of nitrogen in nitric oxide is the guanidino group of arginine, while the oxygen in nitric oxide is sourced from the molecular oxygen that binds to the ferrous ion of the oxygenase domains of nOS. The entire process of sequential monooxygenation requires 2 oxygen molecules and 1.5 molecules of NADPH. Like cytochrome P450, nitric oxide synthase is likewise inhibited by carbon monoxide. Substrate binding, in this case, the binding of arginine, is not required by the nitric oxide synthase to initiate the reaction cycle. Subsequently, the binding of substrate does not alter the reduction potential of the heme. As a recap, when the substrate arginine is present, which is usually the case, it is monooxygenated by the nitric oxide synthase to citrulline with the release of nitric oxide. However, if the substrate arginine is not present, the electron transfer to the heme can still activate molecular oxygen, which will be released as a superoxide. As you can very well see, if the substrate is not present but the nOS is active, potentially destructive reactive oxygen species may be formed. This is one of the endogenous sources of reactive oxygen-nitrogen species in the body. These reactive oxygen or reactive nitrogen-oxygen species are deleterious in the body, as you have learned in the lecture of oxygen metabolism and toxicity. It can damage cellular, mitochondrial, and nuclear membranes, oxidize proteins, and disrupt cellular functions and even cause cell death. Most diseases in the body are caused by oxidant damage brought about by these reactive oxygen species like superoxide. Here's but a few examples of the myriad list of diseases caused by oxidant damage. Let us now look into the different isoforms of the nitric oxide synthase, or simply referred to as nOS. Different members of the nOS family are encoded by separate genes. There are 3 known isoforms in mammals, 2 of which are constitutive and the third is inducible. Let us now examine more closely the different isoforms of nOS. These are the 3 major isoforms of the nitric oxide synthase. The first in the list is Neuronal, also known as nitric oxide synthase 1, or simply as nNOS, the N referring to neuronal. The second is the Inducible or nitric oxide synthase 2, or simply as iNOS, the I referring to inducible. And lastly, the Endothelial or nitric oxide synthase 3, or simply as eNOS, the E referring to endothelial. Let us discuss the first of these isoforms, the neuronal nitric oxide synthase, or NOS1 or nNOS. The neuronal nitric oxide synthase is primarily in the skeletal muscle as a mediator of contractile force, and in the neurons of the central and peripheral nervous systems as a neurotransmitter. In this form, NOS1 or nNOS is a soluble enzyme, although it is localized near the membrane via protein-protein interactions with a host of regulatory and localized proteins. Additionally, nitric oxide synthase 1 is constitutively expressed, not normally induced at the transcriptional level, and is activated by influx of calcium plus 2 ions to increase calcium calmodulin binding activity. As calmodulin is not bound to the enzyme at intracellular basal calcium levels, NOS1 requires an influx of calcium through voltage-gated calcium channels in the sarcoplasmic reticulum in order to raise the calcium concentration, allowing calmodulin to bind, thus activating nitric oxide formation. Nitric oxide is produced generally by NOS1 in the postsynaptic neuron that diffuses back across the synapse to the presynaptic neuron. Nitric oxide synthesis is regulated by influx of calcium through receptor-dependent channels following postsynaptic stimulation of N-methyl-D-aspartate receptors by glutamate. The nitric oxide that is produced by NOS1 in the postsynaptic neuron diffuses back across the synapse to the presynaptic neuron where it will act on the soluble guanylate cyclase. The presynaptic soluble guanylate cyclase, once activated by nitric oxide, produces cyclic GMP, which regulates the synthesis of neurotransmitters norepinephrine and glutamate, thus enhancing further nitric oxide production. It is worthwhile to note that nitric oxide is synthesized on demand, is not stored in vesicles, and freely diffuses across membranes. Its action is not terminated by degradation or reuptake, but removed by interaction with its target, which is an intracellular messenger. Nitric oxide produced by neuronal nitric oxide synthase, or nNOS1, is responsible for various physiological effects. Nitric oxide produced in the central nervous system has been implicated for neural signaling, synaptic plasticity, neurotoxicity, learning and memory, and the perception of pain. Let us now look at the role and functions of the nitric oxide synthase 1 in the peripheral nervous system. Nitric oxide is produced by nNOS1 in the myenteric neurons of the gastrointestinal, pulmonary, vascular, and urogenital systems when activated by influx of calcium. NOS1 is activated in these systems by the influx of calcium through voltage-gated channels. It diffuses to neighboring muscle cell, causing activation of soluble guanylate cyclase. This relaxes the smooth muscle via the same mechanism for vascular smooth muscle. In the GI system, nitric oxide is involved in gut motility and control of the pyloric sphincter. Nitric oxide is involved in the relaxation of the pyloric sphincter to allow the efflux of gastric contents into the duodenum. However, in the pulmonary system, nitric oxide mediates the main bronchodilator pathway. It promotes vasodilatation of pulmonary vessels, thus decreasing pulmonary arterial pressure. In the vascular system, on the other hand, nitric oxide is involved in the neural control of the vascular system, especially in the control of cerebral blood flow. And lastly, in the urogenital system, nitric oxide is involved in urethra and bladder control and penile erection. To continue, nitric oxide synthase 1 is targeted to the sarcolemma of the muscle due to its association with alpha-syntrophin, a member of the muscle dystrophin complex. Thus, the effect of nNOS1 on skeletal muscles is that it is involved in the mediation of contractile force, innervate developing muscle, and perhaps influencing glucose uptake. A study by Chapman and Hall revealed the effect of knocking out the gene for nNOS1 in mice. It has been shown that the effect on these mice is the exaggerated constriction of their pyloric sphincters, resulting to distension of the stomach. Thus, it has been shown that nitric oxide synthesized from neuronal nNOS is needed for the relaxation of the pyloric sphincter to allow the egress of gastric contents into the duodenum. Application of this is the possible use of nNOS1 among infants with congenital hypertrophic pyloric stenosis by promoting relaxation of the sphincter. Another result of the study is that these mice seem to be resistant to brain damage as a result of ischemic injury, suggesting involvement of nNOS1 in this injurious process. On the other hand, it has been postulated that excessive production of nitric oxide might lead to cytotoxic effects on neurons and may cause strokes. Other clinical aspects of nitric oxide synthase 1 is the possible direct application of nitric oxide gas for the following. For hypoxia, nitric oxide can improve arterial oxygenation and decrease pulmonary hypertension. For acute respiratory distress syndrome, or ARDS, that play important roles in pneumonias in SARS and COVID-19, nitric oxide synthase 1 can increase pulmonary vasodilation, decrease pulmonary arterial pressure, and thus improve oxygenation. It can also help improve the cardiac index. Published findings from the 2004 SARS coronavirus infection suggest the potential role of inhaled nitric oxide as a supportive measure for treating infection in patients with pulmonary complications. Treatment with nitric oxide reversed pulmonary hypertension, improved severe hypoxia, and shortened the length of ventilatory support compared with match-controlled patients with SARS. The beneficial roles of nitric oxide in the treatment of COVID-19 pulmonary complications is now being actively investigated. Let us now move on to the second isoform, the nitric oxide synthase 2, or known as the Inducible nOS or iNOS. NOS2 or the inducible nOS or iNOS is found primarily in activated neutrophils and macrophages, astrocytes, and hepatocytes involved in early immune response. It is a soluble enzyme found in the cytoplasm. To continue, as its name implies, NOS2 or inducible nOS is induced at the transcriptional level by cytokines like interferon, interleukin-1, and tumor necrosis factor alpha, or endotoxins as in lipopolysaccharides. This induction occurs over several hours via various inflammatory pathways. In contrast to the two other isoforms, NOS2 enzyme is always activated once synthesized. Calmodulin is already bound to NOS2 under basal physiologic conditions and is thus independent of calcium ion influx. It synthesizes nitric oxide in the nanomolar range, approximately 1,000 times greater than that of either NOS1 or NOS3. In acute inflammation, nitric oxide produced by NOS2 acts as a potent cytotoxin to destroy pathogens engulfed by neutrophils and macrophages such as parasites, microbials, fungi, and even tumor cells. Targets of nitric oxide synthesized by the inducible nitric oxide synthase are metal-containing heme proteins such as cytochromes P450 and iron-sulfur proteins such as aconitase and mitochondrial complexes 1 and 2, which will all be inhibited. Nitric oxide also reacts with thiol groups and tyrosines, causing nitrosation or oxidation of proteins, and with oxygen or superoxide, forming the highly reactive nitrogen and oxygen intermediates like peroxynitrite. This is a diagram of the respiratory burst wherein a neutrophil is shown phagocytosing bacterium and killing it with various reactive oxygen species including peroxynitrite, which is synthesized by the inducible nitric oxide synthase or iNOS as indicated here. In acute inflammation, bacterial endotoxins such as E. coli and other gram-negative bacteria induce the NOS2 gene to produce high levels of nitric oxide to damage pathogenic DNA and inhibit respiration. Nitric oxide, however, cannot discriminate pathogenic DNA from host DNA. Overstimulation of NOS2 induces cell and tissue damage, leading to septic shock in the course of bacterial infections. Overproduction of nitric oxide has been implicated in septic and cytokine-induced circulatory shock. Too much nitric oxide leads to massive systemic vasodilatation and a precipitous and sometimes fatal drop in blood pressure. And finally, let us now proceed to the third isoform, the nitric oxide synthase 3, or known as the endothelial nOS or eNOS. Nitric oxide 3 or the endothelial nOS, or also known as eNOS, is found primarily in vascular endothelial cells lining all blood vessels and cardiac myocytes as well. Like NOS1, it is constitutively expressed and activated by influx of calcium ions. The amount of nitric oxide synthesized in the active state, however, is very low. NOS3 is a soluble enzyme but is found localized at the plasma membrane by protein-protein interaction in the plasma lemma calvulae. So in contradistinction to the other two NOS isoforms, NOS3 is the only isoform that is membrane-bound. Nitric oxide synthesis is activated in response to an increase in calcium ion influx following binding of ligands like acetylcholine, bradykinin, histamine, or insulin, or following shear stress in the blood vessels. Nitric oxide diffuses out of the endothelial cell into the adjacent smooth muscle cells to bind and activate guanylate cyclase to synthesize cyclic guanosine monophosphate. Cyclic GMP then activates protein kinase G to phosphorylate the L-type calcium channels and receptors. This will in turn lead to the closure of calcium channels in the smooth muscles. This in turn will lead to inhibition of calcium influx into the smooth muscle cells, diminishing development of smooth muscle tension and thus vasodilatation. Nitric oxide has several physiologic effects in the vessel endothelium, namely, first, it serves as a vasodilator of vascular smooth muscle. Second, it is antithrombotic by inhibiting platelet aggregation. And lastly, it is also anti-inflammatory by inhibiting leukocyte adhesion and aggregation. For the clinical aspects of nitric oxide synthase 3, these are the anti-atherosclerotic properties of nitric oxide. First, it prevents adhesion of monocytes or leukocytes to the endothelium. Secondly, it inhibits platelet aggregation at the site of intimal damage. Third, it inhibits proliferation and migration of smooth muscle cells. And lastly, it decreases the permeability of the endothelium to lipoproteins. Thus, it is worthwhile to note that nitric oxide synthase 3 plays a critical role in maintaining a basic vasotonus in the hemodynamic regulation such that a decrease in the production of nitric oxide results in hypertension, thrombosis, or atherosclerosis. Nitroglycerin as a source of nitric oxide reduces vascular resistance and hence blood pressure and dilates the coronary arteries, allowing more oxygenation to the heart. It is used for the treatment of angina pectoris, but it was only since the discovery of nitric oxide that its mode of action was deduced. As mentioned before in the mechanism of action and degradation of nitric oxide, there is an enzyme that is responsible for breaking down cyclic GMP that forms in response to increased nitric oxide concentration. There is an isoform of phosphodiesterase in vascular smooth muscle that is cyclic GMP dependent. The type 5 isoform of this enzyme, abbreviated as PDE5, is found in the corpus cavernosum of the penis and in vascular smooth muscles. Inhibitors of cGMP-dependent phosphodiesterases, by increasing intracellular cGMP concentrations, enhance smooth muscle relaxation and vasodilation. Sildenafil is specific for cGMP found in the corpus cavernosum, vascular smooth muscles, and gastrointestinal smooth muscles. Thus, sildenafil inhibits phosphodiesterase 5, delaying cyclic GMP degradation in the corpus cavernosum and thus prolonging erection. The somewhat selective vasodilatory actions of phosphodiesterase 5 inhibitors have made these compounds very useful in the treatment of male erectile dysfunction. Sildenafil, or Viagra, a drug for erectile dysfunction in males, is a potent inhibitor of phosphodiesterase 5. However, the therapeutic indicator for sildenafil is not only limited to erectile dysfunction. This is also approved for the treatment of pulmonary hypertension. However, just like most phosphodiesterase inhibitors, sildenafil can be a double-edged sword because of its potential untoward effects. Because it also inhibits PDE5 in vascular smooth muscles, it can cause a mild reduction in blood pressure due to vasodilatation, which may be clinically insignificant. However, for patients concurrently taking nitroglycerin and other vasodilators, sildenafil can greatly exacerbate this drug's effects, which can potentially cause fatal hypotension. Thus, sildenafil should only be used upon doctor's advice and upon prescription and contraindicated in patients using nitrodilators. Sildenafil is highly selective for PDE5 but also inhibits to a lesser extent PDE6, which is localized in the retina photoreceptors. Thus, a side effect is a blue-green tint in the patient's vision. This ends the presentation on the metabolism of nitric oxide. Thank you very much. Stay safe always.